Now we're going to talk about some more complex cross-pollinations or breeding situations. And the first thing we've been talking about all along, something known as a monohybrid cross. A monohybrid cross is a cross between two individuals concentrating on one definable trait. And we had looked at the purple people eaters with their horns and other situations where we were just looking at a single trait with two individuals. The trait was controlled by one of two alleles, and each of which was contributed by a parent. Now the second step is, and this is what we were looking at, and I showed this as an example of a Punnett square. Now, as I said, things can get more complicated. Again, if you look at two individuals, but now you're looking at two traits, and each of those traits separately could be looked at with a Punnett square as shown here. But if you put them together, the Punnett square now looks like this, which is quite a bit more complicated. And here you have a situation where you, instead of a single letter here, you have two letters for each possible gamete. And in this case, you have one small c gamete is homozygous for this parent, but the big P is heterozygous. And so for each gamete, you can have one of these with this, you can have one of these with this, on the other side you can have one with this and one with this. And so it gives you this much larger, more complex Punnett square. And so you can see that there goes from 4 cells to actually 16 cells. And this is what's known as a dihybrid cross. Now let's change and look at something that's very important in human genetics and Mendelian genetics, and that is, although it was not described by Mendel, most of the work on this was done by Thomas Hunt Morgan, which the Thomas Hunt Morgan Biology Building at the University of Kentucky is named after him. And he was actually a student at the University of Kentucky, but he did almost all of his work in Columbia. And autosomes, or an autosomal trait, is a trait that's carried on chromosomes that do not determine the sex of an individual. So in humans, we have 23 pairs. 22 of those 23 pairs are non-sex related or autosomal, or autosomes. The sex chromosomes are the chromosomes that determine the sex. And here is the typical male or female adult human with 2X, XX, and then the father or the female or the male human with the large X and the small Y. And if the Y chromosome is passed on like this, then the child will be male. If the X chromosome is passed on, the child will be female. Now the female has no choice. It can only pass on an X chromosome. So the general genotypes are either XX or XY, and there are 50% possibility for each. But in a female, it's 100% possibility that this is what exists. In a male, it's 100% that this is what it exists. Well, sex-linked genetic traits are traits that, in addition to being this carrying the gender of the individual, they carry other traits, and those are called sex-linked traits because they travel together with the sex of the individual. One example is the eye color in fruit flies. As it turns out, Dr. Morgan did almost all of his experiments in fruit flies, and the reason he did that was because fruit flies reproduce very rapidly and they have a very short lifespan. They're very easy to keep alive. And so he could do many generations in a very short period of time. Now, as with other traits, you can either be dominant or recessive, and here the eye color is either red or white, and it turns out that white eyes are recessive, so they're represented with little r's, and red eyes are represented with a big R. And it also turns out that this eye color is carried on the X sex chromosome. So if a male has a big R, they're red-eyed, and a little r, there's no other over here, so one little r means that it's a wide-eyed male. But for a female, both X chromosomes have to have little r's. If either of these is a big R, then the female will be red-eyed. And you can see that's shown right here. And a Punnett square looking at this would take these two gametes from the male, this one and this one on this side, excuse me, and these two gametes from the female, and you obviously could have two X's, one with a big R and one with a little r, two X's, one with a big R and one with a little r. So these would both be females that are homozygous for red eyes, or excuse me, heterozygous for red eyes, but they would both have red eyes as a phenotype. And then this male would be X little r Y, so he would be wide-eyed. And this male would be X little r Y, and he would also have wide eyes. And so that's how it works with sex-linked chromosomes. So let's do a couple of examples just like we did before. And we'll do it on this piece of paper. Let's look at the first one here. In humans, hemophilia is a sex-linked recessive trait. If a female is a carrier for hemophilia and she marries a male who is normal, answer the following questions. Okay, well let's do our Punnett square on this. So we have a Punnett square looks like this, and we have a female, and she is a carrier, which means that she has one normal and one hemophilia gene, and a male that's normal. And so if we put the male over here, it will look like this. We put the female here, and it looks like this. And so the four offspring look like this. Okay, so here's our genotypes. We have one female that is homozygous normal. We have one female that's heterozygous or a carrier. We have one male that's unaffected or normal. Excuse me, he's a male. And we have one male who's affected. So this is an affected individual. This one's sick. So the first question is what fraction of female children will actually have hemophilia? Well, here are your two female children right here, and neither of them will have the disease, so the answer is 0. The second question is what fraction of the female children will be carriers? Well, out of the two, there'll be one carrier, so 1 out of 2, or 50%, will be carriers. What fraction of the male children will have normal clotting? 1 out of 2 again. So 1 out of 2 will be normal males, and 1 out of 2 will be affected males. Okay, let's take a look at this second problem. Very similar. In cats, the allele big B produces a black color. Little b produces a yellow color. Notice we didn't change this to a Y. And big B is dominant, little b is recessive. Big B and little b are sex-linked. So if you cross a heterozygous black female with a yellow male, let's answer these questions. Let me move this over just a little bit so I can read it easier. Okay, so let's first draw our square. Make it big enough. And we have a heterozygous female with a yellow male. That's what it looks like right there. So let's put the male over here and the female up here. So you end up with a female that has a big B and a little b, a female that has a little b and a little b, a male that has a big B and a Y, and a male that has a little b and a Y. So the first question is, what percentage of their offspring will be yellow? Okay, so this one will be yellow, and this one will be yellow. So one of the males and one of the females will be yellow. So 2 out of 4, or 50%, will be yellow. What percentage of their offspring will be black? Well, the other two are going to be black. This one is a heterozygous black, and this is a male that's black. So 2 out of 4 are black, or 50%. What percentage of the offspring are carriers? So that's a female who is heterozygous, and here's the female that's heterozygous. So out of the 4 offspring, 1 will be a carrier, or 25% will be a carrier. So a carrier is a heterozygous for the trait, and a carrier is almost always a woman. Now, genetics has always been a little bit more complicated, but here's a couple of terms that you need to understand, and I alluded to one of these 2 terms previously, and that was the concept of codominance. First of all, if a single trait is the result of multiple genes acting together, then that trait is called polygenic, or polygenic inheritance. And there are examples of skin color, for example, is a single trait in which there are multiple genes that interact in order to produce that trait. Neither of these pictures represent polygenic inheritance. Incomplete dominance, I said codominance, I meant incomplete dominance. Incomplete dominance is when a trait is a result of a blending in the heterozygous state. So for example, as we said before, if something is incomplete, if you have a heterozygous now they changed letters here, so that's not right. They shouldn't have done that. But this is big R and little r. So this pink is a result of one being coded for pink and the other coded for white. This is coded for 2 white. This is coded for 2 red. This is homozygous red, the dominant, homozygous white, the recessive, and then heterozygous is a blending, or pink. So that's what's known as incomplete dominance. The third is over here, and that's called pleiotropy. Pleiotropy is when a single gene impacts multiple traits. So you have a single gene and it impacts more than one trait. So it's kind of the opposite of polygenic inheritance, in which you would have multiple genes impacting a single trait. Here you have a single gene impacting multiple traits. Now, epistasis is when one set of genes actually affects the expression of another set of genes. So here we have one set of genes, and then this is the second set of genes. And let's just say that this set of genes affects the coat color of this dog, and this set of genes affects the nose color. And what you see here is if the dog is yellow, then if he has this, he has a whatever color that is, and if he has either one of these, he has a black nose. On the other hand, if he happens to be dark, and they've actually shown this almost as a little bit of incomplete dominance because the heterozygous is a little different than the homozygous, then if he's recessive, he actually has now a black nose, but if he's dominant, he has a white nose. So what epistasis is that this determines how this is expressed. So one set of genes determines how the other is expressed. Now, the other example is shown over here in blood typing, a genotype that has more than 2 options. You know, we commonly talk about tall versus short, red versus yellow, red versus white, etc. But in blood typing and some other traits, you can actually have more than one. You can have A, B, or AB, or O. And each of these is different. So there are multiple, in this case, 4 different alleles. Actually, 3 alleles. O represents the absence of either A or B. And so that determines the blood type. And this is also an example of codominance, which is when neither dominant trait is dominant over the other. It's only dominant over the absence. So A, when it's with O, is dominant, and the phenotype is type A. But when A is with B, the phenotype is AB, and that's codominance. Excuse me. Whereas if you have B with O, then that's type B, and then 2 B's together are type B. So that's what's meant by codominance. And the only time you have O is if you have the absence in both situations. A little bit more about blood typing. Blood typing affects the expression of these little molecules on the surface of red blood cells, and those are called antigens. Antigens are proteins that trigger the blood to produce antibody. And so if a person is a blood group, if B antigen is introduced into the system, then they make antibodies to B. Conversely, if a person is in a B blood group, they make antibodies to A. People with AB don't make antibodies. And people with O make antibodies against both. So if this person is transfused with B blood, their antibodies will attack that blood and destroy it. Conversely, if this person is transfused with A blood, their antibodies will attack. This person is called the universal recipient because they don't have antibodies against either one of these. And then lastly, this person is called the universal donor because they don't express outward antigen. However, if you give any of these to this person, they will attack it. So the antigens present are none, so they can donate to others. And every blood bank has 2 units of type O blood available in case somebody comes in and needs a transfusion and cannot be typed in time. Now, here's an example of what's known as a genetic disorder. First of all, I said before, autosomal inheritance means that the inheritance of the trait is not on the sex chromosomes, but on the autosomes. A genetic disease carrier is a heterozygous person for that genetic disorder, and the genetic disorder is a recessive disorder. Now up here is an example of the 2 types of chromosome change that can occur. It's a situation where the chromosomes change either in structure or number. So here is an example. These are 2 non-paired chromosomes, so they're different. And what happens is that a piece of this chromosome translocates to this chromosome. And at the same time, a piece of this chromosome translocates to this one. So it's change in structure, known as a balanced translocation. So this is just an example of a chromosomal change in structure. This one is an example of a chromosome change in number. Notice the circle here. Normally there are 2 of each, but here you have 3. So this is called trisomy, meaning 3, 13, indicating the chromosome that's affected.